So this is lecture video 3 of the reproductive system. So before you actually view this video, make sure that you have watched parts 1 and 2 of this set. So we're going to be talking about meiosis in this case. So if you recall from the very beginning, we actually talk about this process called mitosis. And mitosis actually occurs in all cells, including eggs and sperm. All right, so all cells in your body undertake mitosis. This is actually how you make 200 million sperm. This is actually how you make, all kinds of like cells, again, skin cells, whatever cells that would be. Okay, cartilage cells. Okay, nervous cells. It's through mitosis. Okay, now in mitosis, what happens is the key point about mitosis is you start with 46. Okay, 46. In this case, not really chromosomes, but actually in this case, 46 chromatids, okay, or in this case single chromatids, which is actually the same as 23 pairs of chromatids, or 23 chromosomes, okay, 23 pairs. So you start with 46, you end with 46. So this is the key point about mitosis. In meiosis, however, this is the big difference between these two. If you just want to think about it this way, is you start with 46, okay, and you end with 46 in this case, okay. What happens is you start with 46, you end still with 23 pairs in meiosis 1, but then at the end, what happens is you actually end with just 23, okay, individual chromatids, okay, not sister chromatids, all right. And just that, not to confuse you, a chromatid is one chromosome from a parent. So that's a chromatid from, let's say, mom. That's a chromatid from, dad. Okay, together they're called sister chromatids, and this is actually called chromosomes in this case. All right, XY chromosome. Okay, 1, and obviously we have they're normally labeled as 1 to 23. 23, the 23rd pair being the sex chromosomes, XX or XY. All right. Now, there are some other interesting things that happen here. So we're going to talk about, meiosis, as opposed to mitosis, undertakes prophase, metaphase, anaphase, telophase twice. So in mitosis, we went through, in this case, PMAT, actually, primarily speaking, MAT, once. In here, we actually undertake PMAT twice. Okay, so there's this another distinction again between mitosis and meiosis. So obviously you have a chromosome between mom and dad, all right? So one from mom, one from dad. One of the, again, so big difference, you start in mitosis, you start with 46 chromosomes, you end with 46, okay, or 23 pairs, you end with 23 pairs. In meiosis, you start with 46, you end with 23. When you have 46 total, okay, chromosomes, that is actually called, you're actually called diploid. Okay. Oh, what happened there? Diploid. And when you end with half of 46, just 23, you're actually called haploid. So in this case, just some technical terms that we need to think about. So meiosis. So let's say again, you start with, let's say chromosome 1, you have 1 from mom. Okay, chromosome 2, you have 1 from mom. Chromosome 3, okay, 1 from mom. You guys get the picture. Let's say chromosome 1, you have 1 from dad, 1 from dad, 1 from dad as well. Okay, so before both mitosis and meiosis, you have to replicate from 46 to 92 first. Okay, so what happens is this gets doubled. So now what happens now is before both meiosis and mitosis, what actually happens is you actually now have 2 chromosomes 1, okay, 2 chromosomes 2, 3 chromosomes 3, okay, you guys get the gist, okay. Interestingly, this is where the differences start. So what happens with prophase 1 in meiosis and prophase 2 and prophase in mitosis are two different things. In mitosis, these two sister chromatids do not associate. They remain separated. However, in prophase 1 of meiosis, these guys actually, if you want to think about, lay on top of each other. So what happens is, I'm just going to make some semblance of laying on top of each other now. What actually happens is they actually lay on top of each other. And when they lay on top of each other, now instead of just two sister chromatids, now you have, right, instead of just one sister chromatid, I'm sorry, now you have two sister chromatids, which is a total of four chromosomes, right? Those 4 chromosomes are called a tetrad, meaning 4. And an interesting happens in prophase 1. This tetrad, ladies and gentlemen, exchange. So what happens is this guy will exchange a region with this guy will, okay, they will exchange regions with each other. Okay, and this is the reason why we have 2 blue-eyed parents. Okay, not me, of course, because I'm not blue-eyed. Okay, and you'll end up with a brown child. I'm sorry, brown-eyed child, not a brown child. Okay, and vice versa. This is one of the reasons why we have 2 brown-eyed parents and we end up with a blue-eyed child. And the reason why that is because of this recombination, or this actual exchanging of genetic material that actually occurs between, in this case, homologous chromosomes. Okay, so again, because of the tetrad. So crossing over, okay, is a incident, is actually a, in this case, an event that is unique to meiosis, specifically to prophase 1. Okay, so now what happens now is, because of that, now you have a mixture of mom and dad in one chromosome. Before, you had a pure mom and a pure dad chromosome. Now, because of this crossing over, now you actually have a chromosome 1 that is a mixture. If you look at this when you have blue and red regions, that has a mixture of both mom and dad. All right, so again, that's one of the variations that actually occurs in prophase. And what happens is, all right, so it still happens. So all the way from prophase 1, metaphase 1, they still line up in the equator side by side like you see here. Okay, and an interesting thing happens now. In another important thing, in metaphase 1 of meiosis versus metaphase, and I'm going to erase all of these things right now because it probably is confusing, right? In meiosis 1, all right, ladies and gentlemen, in meiosis 1, as you can see, the actual chromosomes are lined side by side. So chromosome 1 with a mixture, chromosome 1 are lined side by side. In meiosis, in metaphase of mitosis, they actually do not line up like that. So in mitosis, actually they line up in a line. So this is different. So this is mitosis. In meiosis, this is how they line up. Okay, you guys understanding me so far? So this is how they line up. In mitosis, this is how they line up. They line up vertical. In meiosis, they line up horizontal. And there's this has very, very important things here, because now what happens is, in anaphase 1, in anaphase of mitosis, okay, if you think about it of this as individual chromosomes, if this is what's happening here, let's say this is mitosis, right? What happens is, in anaphase 1, they separate. One goes this way, the other one goes the other way, right? And then eventually, right, and so this one goes this way, the other one goes that way. And then what happens in telophase eventually is these two eventually join together in mitosis to create identical chromosomes. And it's the same thing on the other side because these are both identical. Now, in meiosis, because they are driven side by side like this, each side does not get the same chromosome. All right, they do not get identical chromosomes. So what happens now is, when they divide, as you can see, each of the 2 brand new nuclei in telophase, okay, actually gets a highly unique pair, which came from genetic recombination, from crossing over in prophase 1. So this is, again, a very, very important thing. So now, because what happens now is, and they do not separate. So before they get separated, now they do not, in mitosis, in meiosis, they do not separate. The sister chromatids do not separate. Now you actually have 23, okay, pair of chromosomes. So at this point, you are technically called haploid, even though you're not really haploid, okay? And the reason why, again, that is because these guys do not separate. If this separate, then you will obviously have 46 chromosomes. So they do not separate after meiosis 1. So something important. So in meiosis 1 and mitosis, just plain mitosis, prophase 1, there's crossing over. During metaphase, they line up in pairs instead of individual chromosomes. Okay, two main distinctions already in meiosis 1. When you hit meiosis 2, all right, because of this, now you only have 23 chromosomes. as you guys can see, what happens is further like another round of prophase, metaphase, anaphase, and telophase happens. And now at the end, instead of having 46 chromosomes, what you actually end up is having 23 individual chromosomes in this case. All right? So very, very important. So in mitosis, main difference, mitosis, you start with 46, right? Obviously during interphase, right? You double that up times 2, you get 92. So at the end, when you have 2 daughter cells, you actually have 246 still. In meiosis, again, very, very important math here. Same, 46 times 2. But since you go through prophase, metaphase twice, and you do only undertake duplication once, right? PMAT twice. I'm sorry. Okay. Running out of time here. What happens is 46. You still have kind of 46 here, although you're in 23 pairs. Okay. And what happens is at the end, you actually get only 23 chromosomes. Now, a very, very important thing here. Now, interestingly speaking, this might seem like you end up with 4 cells. In spermatogenesis, that is true, you actually end up with 4 cells. In actual oogenesis, the creation of ovum, you actually only end up with 1 cell, and we're going to be talking about that soon. Now, something very, very important, okay? All of the cells in your body has 46 chromosomes. Sperm and egg cells only have 23 chromosomes. This, if a white blood cell interacts with either your egg or sperm, your macrophages, okay, will actually recognize this as foreign because it doesn't have all 46 chromosomes. So what it does is you actually get your macrophages digesting, eating your sperm cells and your actually egg cells. This is the reason why ova, eggs, are inside a follicle. The follicle prevents white blood cells from interacting with the very, very aberrant, very abnormal number of chromosomes. Because again, if white blood cells can actually detect this, you actually will, in this case, attack your own sperm and your own eggs. In the testis, similarly speaking, there's actually a testis blood barrier. And again, the reason why that is it prevents white blood cells from interacting with sperm. Because again, sperm only has 23 chromosomes as opposed to a normal, all the other cells in your body has 46. So if a white blood cell, especially a macrophage or a lymphocyte, interacts, okay, with an egg or a cell, it'll count its chromosome and says, uh-oh, this cell only has 23 chromosomes. It is abnormal, therefore it needs to be finished off. It needs to be attacked, removed, destroyed, okay, from circulation. Because that is actually a sign of your own cells thinking that, oh, it's a cancer cell. Okay, so this is again one of the reasons why eggs are in a follicle and why there's a testis blood barrier. Okay, so to sum, the main differences is in meiosis you undertake prophase, metaphase, anaphase, telophase twice, only undertaking duplication of chromosomes once. That means to say that at the end, your cells that you actually create only has 23 chromosomes. In mitosis, because you undertake one duplication on only one round of cell division, PMAT, okay, prophase, metaphase, anaphase, and telophase, your daughter cells still have 46 chromosomes. So that is one of the actually biggest difference. Other differences is in prophase 1, in prophase in mitosis, there's no crossing over between chromosomes. In prophase 1 of meiosis, there actually is crossing over of chromosomes. Okay, that actually gives you genetic variability between children. This is why some of your kids don't look the same. Okay. Another important thing is in metaphase 1. Okay, in metaphase 1, as opposed to metaphase of just mitosis, in metaphase of mitosis, the actual chromosomes actually line up horizontal individually. Okay, in this case, horizontal, vertical individually. In terms of meiosis, in metaphase 1, in meiosis, they actually line up in pairs vertically. Okay, this is actually how you only get 23 pairs on each side. And this is actually how you get 4. All 4 of your sperm will become different. And all 4 of your eggs, if you get 4 eggs, actually are genetically different from each other.